Okay. So yeah, as I just mentioned, this is the first chapter where we're actually getting into anatomy and physiology content. And so addressing that term, right, the name of this course is Anatomy and Physiology, which is referring to structure and function. Anatomy is the structure, and then function, or the physiology is the function of those structures. And a pattern that you will see come up again and again, especially if you are aware of it from the beginning, which is why I'm trying to say it now, is that structure dictates function. There's a lot that you can determine about the function of a thing, of a cell, of a tissue, of an organ, based on the structure of that thing. And so I will look for as many opportunities as possible to point out those types of things and give practice with those types of things, just kind of analyzing structure in order to determine general function. And so getting started with that, we are discussing the various types of tissues that build up the organs in the body. And so we kind of addressed generally the organization of living things in the past chapters, right? But we have atoms, which are the basic unit of matter that take all things that take up space in this universe. Atoms form bonds together to make molecules. Molecules can form interactions to create our cells. And then cells will become specialized in larger organisms like ourselves, where we have trillions of cells that make up our body. And so we have cells that specialize for particular functions. And groups of cells that share a particular function are what we refer to as tissues. So a tissue is going to refer to those cells that share a particular function, as well as the fluid that fills the space around that area that makes up the tissue. And then various tissues will come together to form organs, and organs work together in organ systems, all together, working together to support the general life of the entire organism. So yeah, we're moving up from cells into our tissues. And then throughout the rest of this course, we'll be doing a deeper dive into specific tissues, essentially. And so of the tissues in the body, we have four main categories. We have our epithelial tissues, our connective tissues, our muscle tissues, and then our nervous tissue. And so we'll have entire chapters on muscle tissue, multiple chapters addressing nervous tissue. We'll have specific chapters on types of connective tissue like bone. But in general, we're only getting but these epithelial tissues, I guess the point here, a lot of our epithelial and connective tissue are showing up in all of our organs or a majority of our organs. So we're only going to briefly address muscle and nervous tissue in this slide deck from this chapter, and we'll go more in depth with them later. And same thing goes for bone, which is technically a type of connective tissue. We're just going to briefly address bone, but we do have an entire chapter on osseous tissue. All right. So our epithelia, or our epithelial tissue, is going to be what is lining all of the parts of our body that are interacting with some environment or some space. So the most obvious would be our skin, right? Which is thick layers of epithelial tissue that interact with our environment and have to endure a lot of friction as they interact with that environment. But I also want you to think about the linings of the space inside our bodies. All of our body cavities or lumens are going to have epithelium lining them as well. So including the linings of our blood vessels, right? Those cavities around our connective tissue of blood, the lining inside of our digestive tract, right? That separates the stomach cells from the actual stomach fluid in the foods that we are digesting, as well as the linings on the exterior of the organs inside of our body. Like lining all of the organs inside of our thoracic cavity to help them with friction and interactions with each other. So again, epithelia is covering all of those exposed surfaces. Anywhere there is a space, there's going to be a lining of epithelial tissue separating the rest of the cells from that space. Connective tissue is what is always under epithelial tissue, essentially connecting it to whatever else is going to be under it, as well as connecting organs to each other, so the external parts of those epithelium. And so these two categories of tissue, being so prevalent and varied in their function and usage, are going to be the most diverse. We have lots of different types of epithelium that we'll be talking about as far as types that have names and have been categorized specifically. The connective tissue, we have maybe a few different categories of connective tissue, but the components that make up connective tissue is the most diverse. We have lots of different types of cells, lots of different types of fiber, and then the ground substance, the fluid that makes up connective tissue. So these are the most complex and varied. And then muscle tissue, we only have a few different types, and we'll talk about those again in their own chapter. And then nervous tissue, we also only have a few different types, but they make up the entirety of our central and peripheral nervous systems, which obviously have very important functions for our body. So we have multiple chapters discussing nervous tissue. Generally, this is just a figure kind of exhibiting the same information that was on that last slide of where these things are most commonly found. But we're going to look into the specifics, and I'm going to try my best to bring some energy to this topic as much as possible. It's very easy to get drawn into just reading off the slides because it is a lot of info, and we're just kind of differentiating between types within these categories. So I'm going to do my best to bring some energy and show some application as we get through this, so it isn't just reading off slides, trying to make that as little as possible. Okay, so for each of our tissue types, we're going to identify some of the common characteristics throughout that type of tissue. So epithelial tissue are unique in that they are polar, and not polar in the sense of how we were discussing it with chemistry, like polar covalent bonds creating charged poles. Polar in the case that we have two very distinct sides, so two distinct poles with very different shape and function within that tissue, within these cells. So we have an apical surface, which is always going to be facing the environment that epithelium is lining or separating against. And then the basal surface, which is where the epithelium are connected to the connective tissue underlining that epithelium. Epithelial tissue is made almost entirely of cells, as you'll see when we move on to our other tissue types. There are multiple protein fibers and different types of liquid that make up the tissue of some of the other tissue types. But for epithelium, it is almost entirely just the cells themselves that are making up that tissue. They are avascular, so we don't have any direct supply of nutrients to our epithelium from our blood vessels, from our lymph. So they are mainly getting their nutrients through absorption from the neighboring cells, from connective tissue, which are vascular. And then our epithelial tissue are all being replicated at a really, really high rate. So since this is the tissue that is separating the internal portions of a tissue or of the organ, separating our cells, I guess, from space, they're mainly dealing with a lot of friction, a lot of opportunity for damage. They're exposed to a lot of enzymes, such as the digestive enzymes through our digestive tract, right? And so we have stem cells, essentially, that are there to generate these cells and replace them at a really high rate. Which also means that the cells of the epithelial tissue are most prone to mutation and thus most prone to the formation of cancer. We haven't really addressed that much, but hopefully you learned about it in biology. Essentially, mutations are most likely to occur during cell replication. So when one cell is forming two new cells and all of the DNA needs to be duplicated and replicated, that is where there is the most likelihood of a mistake being made in the genetic code. And those mistakes can be fixed, but if they get left behind and those mistakes start to build up, eventually those cells will no longer perform the function they were supposed to, and often will be replicating at a higher rate than they're supposed to, and that's what forms a tumor or cancer. And so since we have such a high level of regeneration in our epithelial tissue, this tissue is where we are most prone to creating mutations and to forming cancer, right? Or at highest risk of forming cancer due to that high rate of division. Okay, polar, made almost entirely of cells, avascular, so we're getting all of our nutrients to this tissue through diffusion, and then continuously regenerating. So here's a look at an example epithelium. This would be columnar epithelium, which we're about to get into the naming in a few slides here. But wanted to point out the polarity shown here. So every cell as a part of an epithelium is going to have an apical surface, which again is going to be facing whatever environment we're discussing there. So again, that can be space within our organs, the lumen, as well as our external environment on our skin. The apical surface is what is pointing out. And then we have our basal surface, which is where those cells, where that tissue is connected to the connective tissue underlining it, right? With a basement membrane. And so this basal surface is mainly collagen fibers, essentially there to just hold those cells to the connective tissue under it, and resist movement and resist stretching. So apical surface faces the space, basal surface connects that tissue to the rest of the body, to the other tissues. And so we have a variety of structures that help to hold these cells to each other, as well as hold those cells to the underlying tissue. And I have a slide looking at closer at all of these portions here, A, B, C, D, E. So we won't go over all of it there. So essentially, we have a few different examples of structures that have come about as forms of adhesion, so holding neighboring cells together. So we gave a name to the apical surface and the basal surface. We also have our lateral surface on the sides, which is what is interacting with neighboring cells. And so on that lateral surface, we have tight junctions, which are super, super tight, do not provide any space for diffusion of molecules in between cells at those locations. So just actin webs, these microfilament webs that are holding those cells together. We have a terminal web of highly densely compacted microfilaments, so more actin fibers, which creates the strongest or some of the strongest connections, and has a very specific adhesion belt along that terminal web, holding the terminal web of neighboring cells together. We have gap junctions, which are full of porous transport proteins, which will allow the diffusion of molecules between neighboring cells. So any regular diffusion or transport of molecules between neighboring cells and epithelium is going to be through these gap junctions. And then the tightest or strongest connections that these cells have are at desmosomes. And we have a few different examples of desmosomes, but in general, it is essentially the cellular glue, super, super tight junctions between neighboring cells on our lateral surface. And then we have hemidesmosomes, which is what connects the basal membrane to the basement, or connects, the basement membrane to the basal surface. Okay, so here is a gap junction. Like I said, it has these channel proteins in here that create space for molecules to move through and diffuse between neighboring cells. And these proteins are called connexons. So they create connections, create a channel, connexons, for small molecules and ions to just easily transport and diffuse between our neighbors. Here's a closer look at those tight junctions and adhesion belts. So again, these are microfilament structures. These tight junctions are essentially like staples, right, holding that together. So no room for diffusion of molecules. The adhesion belt has its own special name because it is where the cells are connected along their terminal webs, which are just long extensions and webs of those microfilament fibers, so very similar. So the tight junctions, but it expands throughout the cell and gives it some structure and holds its shape. We have at our desmosomes a lot of these special cell adhesion molecules. And this is kind of what makes it that super glue of the cell, these cell adhesion molecules, which are proteins and carbohydrates that work together to create really strong connections, right? That's super glue. And on the external portions of these desmosomes, we have an intermediate filaments network that again also provides structure and help for these cells to stay together. And then lastly, just another example of a desmosome are hemidesmosomes, which is what connects the basal surface to the basement membrane, and then the connective tissue that would be lying under it. Right? So we would have cell adhesion molecules as well as some other protein fibers here, just connecting it to the underlying tissue, preventing it from just floating away into whatever space it is lining, and preventing stretch and movement of the cells. So all of our epithelial cells are going to have this basic structure, right? What we're looking at here is an example of columnar epithelium. It has that column shape. And this would very likely be the columnar tissue that you would see in like the linings of our small intestine, which have our microvilli, those actin extensions, microvilli, to increase surface area and allow for more absorption of the nutrients that we're digesting. And so these structures we've addressed are going to show up in all of our epithelial cells, even if they don't have this exact shape. Right? So we do have a few different shapes, and that is what gives our different categories of epithelium. So epithelia are classified by and named by their shape and their layers. And honestly, I wish I'd switched this because you name them with layer first and then shape. So stratified squamous epithelium. We're naming the layers, which is stratified, meaning there's more than one layer, and then the shape. This is a squamous shape. And so we're going to look at the examples of all 6 of the types that you see here. And there's a couple others that aren't on this list just because they are a bit unique in how they are structured to give them their names. And we'll talk about examples of where they show up. But again, in general, I'm going to try to avoid just listing organs of where you find these and talk more about like the application of why this type of epithelium would be beneficial and thus the areas you would be more likely to find it. Right? So trying to connect that logic portion of the structure and function. So shape and layers. We have 3 different main shapes. We have squamous, which literally means scale. And so these are going to be really, really flat. So not much space within that cell. And thus they are biologically inexpensive. And that's a phrasing that you may hear more and more through this class. When I'm talking about biologically expensive versus biologically inexpensive. Essentially, for a living organism, if nutrients supplies are the currency, how expensive a cell is going to be based on how many nutrients supplies it's going to take, right? Your body only has a certain amount of energy to offer, a certain amount of building blocks to offer based on your diet and what you're consuming. And so these simple squamous cells are going to be our least biologically expensive example of tissue here, right? Really, really thin, not a whole lot of space in them, not a whole lot of structure. And so these are present mainly where there is friction. And so simple, meaning there's only one layer, and stratified, meaning there is anything more than one layer. Our simple squamous epithelium is going to be in locations where there is friction, but not rough friction or not a whole lot of friction, such as the spaces that are lining, like touching organs, such as the organs in our thoracic cavity. Right? We have organs on top of each other, essentially rubbing, that have the potential to rub against each other. And so we would have simple squamous epithelium on that external lining to help with that friction, and they can easily be replaced and aren't very expensive to replace. Our stratified squamous epithelium is going to be in places where there is a lot of friction. So again, generally inexpensive cells because they don't take up a lot of space, don't require a whole lot of structures or energy to build them. And we can stack them on top of each other to provide more layers of protection. And so where there's a lot of friction, places like our skin, the linings of our mouths, the linings of cavities that interact with our external environment often, you're going to find stratified squamous epithelium. So reducing the probability that the connective tissue that is underlying this epithelium would be damaged from that friction. Okay. Next, we have cuboidal. So a bit more shape. These are named cuboidal because they generally are the same length along all sides of this 3-dimensional cell. They're not always exactly cubes, but generally in this kind of cube shape. And they can be simple or stratified. So we have a bit more space. So these are typically going to show up because there's more space, there's more room for functions. And so cuboidal epithelium typically shows up where there's a little bit of secretion. So like our glands that need to secrete hormones or fluids are mainly going to be some form of cuboidal epithelium. So again, we have more space in the cell. There's more room for actual processes of the cell to occur, so the production of secretions like hormones. And so these cuboidal epithelium show up mainly in glands. And then lastly, we have our columnar epithelium. So they have almost a column shape, almost rectangular, I suppose, right? They're very long, and they again can be simple or stratified. And since we have even more space here, now these are the cells that are most likely to be involved with absorption and secretion. So when there is something that needs to be secreted in high volumes, such as the mucus that protects the internal environment of our stomach or protects the cells of our stomach. Right? So any area where there is going to be a lot of secretion or high volume of secretion is most likely to have a columnar epithelium lining it. So there's the most room for cellular processes to occur. Okay, hopefully that's helpful. And so now we're going to move through all 6 of these are simple and stratified versions of each. And then there's a couple other examples. There's transitional epithelium, there's pseudo-stratified columnar. And again, it's listed where these things are. So for instance, looking at simple squamous epithelium first. So again, this is one layer of those really flat scale-like cells, so relatively inexpensive cells. These are going to be lining areas where there is a bit of friction in order to provide that protection. And so you could expect to find these in areas where organs are rubbing against other organs. So not a whole lot of friction, at least compared to like our skin and the external environment, but there is some friction. And that is where you would most likely find simple squamous epithelium. I also added these icons, these I haven't had until this quarter, with the hopes that that's helpful in quickly flicking through these and finding examples of where they are. It's based off of this. But yeah, keep a lookout for those. So this is our most delicate because they're thin, located in these slick areas with a little bit of friction. Again, like linings of places like the alveoli of the lungs, linings of our thoracic and abdominopelvic cavities. So those are the spaces within our body where organs are rubbing against each other. And then the linings of our vessels of the heart and blood. Okay. Stratified squamous, meaning we have layers of these thin scale-like cells, is going to be most useful in areas where there's a lot of friction. So protection from that external environment, such as our skin and our mouths, the linings of the esophagus, rectum, anus, and vagina, right? Areas that interact with moving parts from our external environments. So we have lots and lots of layers. So if we experience friction and some of those layers get rubbed off, it's easy to replace them. And we're still protecting the tissue that is under them because we have lots of layers. Well, we have a whole chapter on skin, but skin specifically has these keratinized epithelium with a lot of that keratin protein, which is really, really tough and makes it water resistant. Not necessarily to prevent water from the external environment getting into our bodies, but to prevent water from leaving our bodies, to keep the water inside of our cells and prevent dehydration. Okay, so now cuboidal epithelium. Again, we have a little bit more space in these cells now compared to our squamous, and so there's room for functions. And so most of our cuboidal epithelium is going to be involved in secretions that aren't being released in high volumes. So things like hormones, or specifically all of our glands are going to be some sort of cuboidal epithelium. So we have simple cuboidal epithelium, which again is part of glands and ducts, portions of our kidneys and the thyroid gland. And then we have stratified cuboidal epithelium, which is rarer, typically only a couple or a few layers of those cuboidal shaped cells. And these are really only found in some of the larger glands, like our sweat glands and mammary glands. And this might be a good time to introduce a new term as well. How do I get into here? There you go. Let me get a text box. Shut up. Let me get a text box here. So speaking of glands, we have endocrine versus exocrine glands, which can be an annoying thing to remember or keep track of. But essentially, if you break this down, endo, a prefix meaning into, and exo, a prefix meaning out of, endocrine glands secrete into our circulatory system. so into protected tissue in the body that is going to travel throughout the body. Exocrine glands are secreting into some space or into the external environment. Right, so our sweat glands are secreting sweat onto the exterior of our skin, or tear ducts secreting tears out of our eyes. Our mammary glands are secreting milk to be secreted out of the body. Right? Those are all exocrine glands, whereas the glands that are producing hormones and the glands that are secreting secretions that are going to go into the lumens of our organs, such as the space inside of our stomach or inside of our small intestine, those are endocrine glands. Or sorry, let me back up, because I was a bit misleading there. Endocrine is always into the circulatory system. So yes, for the hormones, but into the lumen and spaces of the stomach and the small intestine, that is still considered an exocrine gland. So secreting into space or external environments, so out of the tissue, exocrine. Endocrine is into the circulatory system. You will see that in the textbook if you're reading through this chapter, but I don't think it shows up anywhere else in this PowerPoint, so I wanted to address that. And so again, cuboidal epithelium is typically what makes up our glands. So they have the room to produce whatever secretion that they are secreting, but it's typically not secreted in super high volumes. Compared to our columnar epithelium, which has a lot more space, and so has the ability to secrete secretions in higher volumes, right? That columnar shape, creating more space. And this is often lining a lot of those lumens that I was talking about, so the spaces inside individual organs, such as the lining of the stomach, intestines, gallbladder, uterine tubes, the kidneys. That's where you're most likely going to find these columnar epithelium that need to secrete into that space or absorb things out of that space. Right? Throughout our entire digestive system, we are breaking down the food that we have consumed in order to absorb it into our cells and then get it somewhere else in the body. But we're also secreting a lot of things into that space to help with the digestion. We're secreting a lot of enzymes to break down all the different nutrients in our body. We're secreting mucus to protect the cells, since we have a very acidic environment in our stomach and a very basic environment in our small intestine. And this columnar shape leaves space for producing and storing those secretions in order to release them. So backing up a little bit, we have our first example of a weird classification of epithelium here with transitional epithelium. So transitional epithelium are almost cuboidal in shape, but they change their shape. So this is in lining or areas that need lining that is able to stretch, essentially, that needs to be able to expand and then recoil. And so these are found in the urinary system. So in our bladder and the ducts and tubes of our urinary system, you could find this transitional epithelium, this unique type here. Again, it almost looks like stratified cuboidal, but they do change shape depending on how full that space is. And they can tolerate those repeated cycles of stretching and then returning, contracting back to where they were, which the other epithelium cannot. Okay, we talked about simple columnar in general. Let me skip ahead a bit here. We have our stratified, so we had our simple columnar lining a lot of those lumens, those spaces. We will also find stratified columnar epithelium in some of our other lumens, some of our smaller areas. It's just part of the spherynx, the anus, the mammary glands, salivary gland ducts, and urethra. So just where there is smaller spaces, they still need to have some sort of secretions and absorption occurring. You would get these layers of those columnar epithelium. And then our last example of a strange, unique type of epithelium is pseudo-stratified. So pseudo, a prefix meaning fake. So pseudo-stratified is referring to the fact that these, if you look under a microscope, will look stratified or are easy to confuse with a stratified epithelium. Even if we look at this image here or the actual electron microscope image over here, they're oddly shaped. They don't keep that almost firm columnar shape that we were seeing before. However, each of these is one single cell whose apical surface reaches the internal space and basal surface is directly connected to that basement membrane. But because we have these weird shapes and they're squished within each other, the nuclei end up in different locations. So if you saw this under a microscope, it'd be very easy to confuse this with 3 or 4 cells stacked on top of each other and assume that this is stratified, that we have multiple layers. But these are each single cells that are completely connected from the apical surface all the way to the basement membrane there. And so you find these in mainly parts of our respiratory system, as well as parts of the male reproductive system. So they also provide secretions mainly with mucus, and are very commonly have these cilia to help move whatever is moving in that environment. Okay, so those are all of our various types of epithelium. Again, without overloading you with, or trying to avoid overloading you with just memorizing each and every organ that these things are a part of, try to focus on that idea of shape determining function. So addressing generally what this shape would provide compared to another shape and the advantage of that, and thus where in the body that would most likely show up. So what we were addressing a few minutes ago. Are there any of these you guys would like me to touch on again or explain in more detail? Either with structure or types of epithelium? Okay. So next, moving on to connective tissue. So this is the most diverse of our 4 tissue types, mainly because of just how many different cell types and how many different fibers and different combinations of fluid versus cells versus fibers make up our connective tissue. But this is the tissue that is always going to be in between your epithelium and the underlying tissue, whatever is the major core part of that organ, as well as connecting different parts of the body. So bone and cartilage are technically connective tissue here. Blood is technically connective tissue. The tendons and ligaments, which connect those things to each other, those are all parts of connective tissue or types of connective tissue. And so this is really the tissue that, this is why we cook our food, I guess is what I'm trying to get to. Like meat in general from animals is quite tender. The actual muscle tissue itself is very tender and easy to chew, easy to rip apart. But it is the connective tissue surrounding it and connecting it to all the different structures that is really, really tough, that we would need to cook, supply heat and energy to break it apart in order to get it to a place where we can actually digest it mechanically and chemically. So it's connective tissue. Again, this is what is connecting epithelium to the rest of the body, not only including bone, cartilage, and blood, as I was saying before, but also fat. Our adipose tissue is a connective tissue. This is never exposed to the internal environment, so it's always between the epithelium and the underlying tissue. They're commonly highly vascular, so whereas the epithelium didn't have any supplies directly from the circulatory system, the connective tissue does. And so the nutrients that get to the epithelium are going to diffuse from the connective tissue to the epithelium. And they also contain sensory receptors. So when you are feeling the things under your skin, you're feeling that through the connective tissue that is underlying the stratified squamous epithelium, which is making up those layers of actual skin cells. Okay. And our connective tissue are all going to have the same 3 basic components. It's just the types and the concentration of each that will vary between our classifications. So you could simplify this even further. Instead of 3 components, you could look at it as 2 components. Essentially, you have your cells, and then you have the matrix surrounding those cells. But the matrix is composed of protein fibers, and then the semi-fluid ground substance. Or I guess depending on the type of connective tissue, this could be extremely fluid or more viscous, but it is a semi-fluid ground substance. So essentially what is filling the space, like the cytoplasm of the cell, that is the ground substance if the organelles are these cells in that case. So again, we're gonna have a few different types, a few different classifications of connective tissue, but all of them will have some amount of specialized cells, some concentration of protein fibers, and then a either viscous or very fluid ground substance, depending on the type. Okay. we've kind of addressed this, but generally connective tissue function to establish our structural framework to help with transports. Blood is a part here, so transporting those nutrients throughout our entire body, lymph as well, our immune system cells. They protect our delicate organs, they support all other types of tissue, store energy, especially with our adipose tissue, which is storing fat cells as long-term energy storage. And they help us defend against invading microorganisms with our immune system cells, our white blood cells that travel through the blood. So our main classifications of connective tissue are connective tissue proper, which just generally has a close to even balance of cells to matrix. We then have fluid connective tissue, which has an extremely liquid matrix and is much more matrix and ground substance than it is cells and connective fibers. And then we have our supporting connective tissues, which are much more densely packed with those fibers, very, very little fluid, or at least extremely viscous ground substance where there is that fluid. And then cells are less important here. So these are our cartilage and bone. And so a majority of the structural component, the dense part of the cartilage and bone, is going to be the fibers or the mineral deposits, in the case of bone, that give it that hard property. All right, so again, connective tissue proper has generally a balance of all three of those things: cells, connective fibers, and the fluid, the ground substance. And so we'll have subcategories of each of these classifications as well, and then subcategories of those subcategories in some cases. So connective tissue proper can be separated into loose and dense, which, as the name suggests, referring to how much space is between the stuff in that matrix. So loose, having a lot more space; dense, having less space. But then we have categories of loose connective tissue and categories of dense connective tissue. So in general, within connective tissue proper, you could find examples of all of the things you see on this slide. We have various different cell types, various different fiber types, and then a few different things that make up the ground substance, depending on what you're looking at. And so I'm not going to talk about every single cell here, just reading the definitions of what they are. But again, I'm going to try to point out some patterns and things that will stick out, hopefully, often. So the first one, the suffix blasts is always referring to some type of stem cell. So any kind of blast, a fibroblast, an osteoblast, those are stem cells that are responsible for producing more cells or more of the ground substance, more of the matrix. So ground substance and fibers. These are stem cells. Anytime you see blast, that is going to be something that is producing more of that tissue. The suffix cyte, referring to cell, is always going to be a mature version of the cell in that tissue. Right? So fibroblasts can mature into fibrocytes. And typically the mature version, the fully formed mature version of these cells, are mainly going to be there for maintenance of the structures within that connective tissue. So the blasts generate all the stuff as stem cells, and then the cytes are within the tissue trying to maintain it. Okay? So fibrocytes are maintaining those connective tissues. Adipocytes are maintaining those lipids. They're essentially cells that have been completely dedicated to, as a vacuole, a space for the fat to be stored into it, the lipids to be stored into it. So mesenchymal cells are just a general category for all the cells that are kind of responding to damage. So these could include stem cells like fibroblasts. These could include the phages. So here's another suffix to talk about. A phage is any type of cell that is going to be responsible for digesting or destroying other cells. So your macrophages are very large immune system cells, white blood cells, that are there to absorb and kill invading bacterium and viruses and fungi. Again, we have cytes here for a mature cell responsible for synthesizing and storing the protein melanin. Just talked about phages. Again, phages are going to be destroying invaders or destroying some cell or protein. And then we'll talk more about specifics of blood cells when we get to that slide. So in general, there's a lot of different types of cells that can be in our connective tissue. They do all have their own specific functions, but if you can start to recognize those pieces of the word and break them down, hopefully every time you see an example of this term, you should generally have an idea. So fibro referring to fibers, cyte a mature cell, which is going to be responsible for maintenance. Fibrocytes are cells that maintain the connective tissue fibers. So hopefully that will start to build. Let me take a drink of water. Okay. The fibers within connective tissue, we have a few different types. Collagen, which is super, super, super tough. Okay, so this is what is packed together very densely in our tendons and ligaments. So tendons are responsible for connecting muscle to bone. Ligaments are responsible for holding bone to bone and sometimes other organs to bone. These are very densely packed collagen fibers. And so collagen is like the rope of our bodies. Super, super strong, like if when pulled from end to end, so directly in the linear direction that it is designed to resist stress from, it is stronger than steel, can resist more force than steel being pulled apart. Very, very strong fibers. But again, they are directional. It can only resist tension from pulling apart. We then have reticular fibers, which are thinner and are more likely to form branching networks. And so reticular fibers will typically show up where there is tension from multiple directions. And then lastly, we have elastic fibers. So whereas the collagen and reticular resist stress or tension, elastic fibers are made to stretch and then snap back to where they were. So these are all made of types of protein. We have elastin, making up elastic fibers, collagen, made of collagen, and reticular fibers. Okay. Skip through images. Okay, so now getting into our subcategories of categories. So we are still looking at connective tissue proper, which is separate from fluid connective tissue and supporting connective tissue. But now we're going to look at the few types that are considered loose and then a few types that are considered dense. So our first example of a loose connective tissue is our areolar tissue. So this is connective tissue that would be found to holding tissues together almost or under almost all of our epithelium. Okay? So all of those lining tissues, connecting that lining tissue to the underlying tissue, you'll have areolar in between. So this is loose connective tissue. There's a good amount of space. We have lots of fibers. So lots of fibers, a little bit of cells, and lots of ground substance. So space in between. Again, this is connecting epithelium to the underlying tissue almost everywhere. Adipose tissue is also considered loose. It is, again, essentially these cells have been specialized to be vacuoles for storing fat, for storing triglycerides, so lipids to be used later. And so those could either be used as energy storage for later, but they also provide protection and insulation. So they resist changes in temperature, as well as providing some padding. And besides resisting temperature, they also protect against electrical activity. So you have adipose tissue around the lining of your heart, around areas where nervous tissue is going to come into close contact with other tissues. We would have a layer of adipose tissue there. And then our last loose connective tissue is reticular. So this is going to be basically anywhere where there is blood kind of holding those vessels in place within those organs. So you can see we have kind of a web of those reticular fibers, right, to resist stretch and tension from multiple directions. So again, areolar tissue holds epithelium to the underlying tissue. Adipose tissue is providing a bit of a buffer in between tissue types, as well as energy storage. And then reticular tissue is what is lining all of our vessels, essentially, holding together areas where fluid is going to be traveling through. So those were all loose connective tissues. These next few slides are going to be dense connective tissues. And as the name suggests, these tissue are all going to be much more densely compact with the structures within them. So we have, and mainly with fibers. Okay? So we have dense regular connective tissue, which looks a lot like muscle. When we get to looking at muscle cells, and a few slides here, they're very long. They have striations like this, but these are collagen fibers. These are not long muscle cells in the way that you would find in a skeletal muscle. These are collagen fibers making up this dense connective tissue. And so this is what you would typically, what a tendon would look like, right? So this is really dense collagen that is going to hold a muscle to the bone, stabilizing the position of those bones and connecting the muscle to it. We then have our dense irregular connective tissue. And so this regular connective tissue has linearity to it. We have end-to-end collagen fibers going in the same direction. So they'll be really good at resisting tension that follows that direction in either side. But our irregular connective tissue has these fibers in bundles in different directions, so it can resist tension in various, stretch in various directions. And then our last dense connective tissue is elastic tissue. So whereas the other two were resisting tension, elastic tissue provides some flexibility. Okay? So anywhere where there is connection in this way, where there needs to be some sort of movement. So most of our ligaments, so where bones connect with bones, right? Those bones are never directly hitting each other. There's almost always some sort of cartilage in between, and then some ligament holding them together, which provides for some fluidity and movement and elasticity. Right? So we have these elastic ligaments between our vertebrae, allowing our vertebrae to move so that we can rotate our body and flex in the way that we need to. Right. Last bit of terminology here for connective tissue is fascia, fasciae, depending on if it's singular or plural. And these are just layers that make up our connective tissue framework. So general categories of where those connective tissue are laying: superficial, deep, and subserous. You may also hear the term fascia being used to describe like essentially scar tissue in the muscles. If you get a massage and you regularly see a massage therapist, their goal is to try to work on this muscle fascia, reduce that buildup of the scar tissue based on how much stress you've put on your muscles. Right? But these are generally just identifying the areas, the layers of our connective tissue. So superficial, deep, and then we have subserous fascia lining again with a serous membrane, which we'll address at the end of this. All right. So again, all of those were technically connective tissue proper. So we branched from connective tissue proper into loose and dense. And then under loose, we had our three categories. Under dense, we have our three categories. And now we're moving on to fluid connective tissue. And so this is essentially our blood and lymph, so our circulatory system and our lymphatic system. And so we have a few different cell types. Obviously, the matrix, the ground substance, is what's taking up a lot of the space in our fluid connective tissue. It's a lot more fluid, very little fiber in the matrix of these tissues. And so we have these cells traveling through that fluid. And so generally, these cells are there to help take air, so take oxygen and carbon dioxide throughout the body, take nutrients that we have digested and absorbed through our digestive system throughout the body, and then provide protection against invaders that might be trying to travel throughout. So again, bacterium or bacterial cells, fungal cells, and viruses. And so our red blood cells, or erythrocytes, are mainly there to transport oxygen and transport carbon dioxide. So we're breathing in oxygen, taking that oxygen to our cells to be utilized for cellular respiration, right? To allow us to use our mitochondria to make ATP, and then taking carbon dioxide back to the lungs to be breathed out. Something kind of unique about red blood cells is they do not have a nucleus, they do not have mitochondria, so they solely rely on glucose as their energy source. We then have our various cell types of white blood cells, which is there to help with defense. So part of our immune system that is traveling throughout. So we talked about macrophages a bit earlier, which are the largest example here. They're to destroy any of those invaders. But we have a few other phagocytes, examples of smaller cells that are there to, again, a phage is there to destroy something. And lymphocytes, which make up our lymph. So not going to get into too many of the details and specifics as far as memorizing the names of these cells and what each of them does differently, but generally recognizing anything with a fill is part of our white blood cells, or is a white blood cell, and then our lymphocytes through our lymphatic system and monocytes. And so our last cell type as a part of fluid connective tissue are platelets. And so this is where the fiber comes in. Platelets are essentially cytoplasm that is traveling around with some fiber in it, which is going to be used to respond to damage and inflammation. And so it will cause a blood clot in that area. This is cytoplasm with fibers embedded into it. They will cause clots by adding a whole bunch of fiber in that space. So mostly fluid in the matrix of our fluid connective tissue, a few different cell types, and then fibers really only being carried by these platelets to be used for clots. Okay. And then our last category of types of connective tissue is supporting connective tissue. And so this is our cartilage and our bone. So these structures are going to be mainly fiber or some extra deposits into the matrix, like with bone. We have a few dispersed cells within there, but not very much fluid for these. So cartilage does have some, but much more viscous fluid compared to our other examples. So cartilage has a firm gel-like matrix, very high in these complex polysaccharides and proteoglycans. These cells that make up cartilage are chondrocytes. So this prefix chondro is referring to cartilage. A cyte being a mature cartilage cell. So we do have chondroblasts, which would be the stem cells of cartilage. And chondrocytes occupy these small little chambers called lacunae within the cartilage matrix. Cartilage is also avascular, so only gets its nutrients through diffusion, similar with our epithelium, and is separated from the surrounding tissue through perichondrium. So we can see what that looks like through these examples. Cartilage replicates and grows through two types. We have interstitial growth, and we have apositional growth. Oops, I don't know what I did there. Interstitial versus apositional. And if you break it down quite simply, interstitial, meaning within or between, interstitial growth is growing the cartilage from within the cartilage. So we have our chondrocyte within its lacuna shell. It will replicate within that lacuna, within that matrix, expanding and depositing more of the matrix, and then they will continue that. And so the cartilage is growing from within what was the cartilage to begin with. It will start to fill up the matrix, the surrounding matrix, from within. So that is interstitial growth, growth from within, compared to apositional growth, which is adding cartilage from an exterior portion of the tissue, essentially. So we have stem cells, which will differentiate into chondroblast stem cells, which will eventually mature into chondrocytes and be deposited into the matrix and secrete matrix. So instead of them growing with it already surrounded by matrix, separated within the lacunae, they are developing within a new matrix and expanding it from this side of the tissue. So interstitial growth, growth from within the matrix, apositional growth, growth by adding onto the outer surface and expanding from there. And this is the perichondrium that we were just talking about, which separates cartilage from the surrounding tissue, which is mainly just going to have those dividing stem cells and some fibers in it. Okay. And we do have a couple types of cartilage, which is going to be something we address again, or you start to see again when we're looking at the joints, or the chapter on joints in this class. So we have hyaline cartilage, which lines a lot of the connections where bones are meeting other bones at joints. It has a very smooth matrix and allows it to be fluid. We have elastic cartilage, which is what makes up like our ears. So the cartilage that you have extending out of your body, essentially, or making up its own structure separate from the bone, like our ears and nose. So very elastic and stretch and then snap back to where it was supposed to be, rather than lining the edges of bones that are connecting with each other. And then we have fibrocartilage, which has the toughest, most fibrous matrix of our examples here. And so these are make up any padding between bones, joints. So within our knee joint, within these discs between our vertebrae here. Right? So very fibrous to resist that pressure and allow for some elasticity. So it allows for some movement, but limits that movement so that it's not too extreme. And then our last type of supporting connective tissue here is bone. And like I said, we'll have an entire chapter on bone where we discuss the ins and outs of it. But essentially, if you zoom in here, you see where they're looking at these darkest spots are the actual cells. So these little brown things that seem like they have webs coming out of them. Those are the actual mature osteocytes. You have osteoblasts, which are the stem cells making those osteocytes. You have osteoclasts. It's a new one that we didn't show up here, but the suffix clast is there to destroy rather than create. So if a blast is a stem cell that creates, clasts are cells that destroy. And so our bones are constantly building and breaking down depending on the needs of the body. And that is because a majority of the matrix here is made up of these hypoxyapatite crystals, which are like calcium, phosphate, and then forgetting the third, magnesium. I don't know if that's correct, but 3 minerals essentially making up the crystal around here. And again, we're going to have a chapter on it, so we'll get into the specifics later. But these are mainly those minerals. And so when our bodies are in need of those minerals, like calcium in particular, we will break down. Osteoclasts will be activated to start destroying the matrix of this bone and thus release more free calcium to be sent throughout the body to where we need it. And calcium is very heavily used for transduction of signals and for the contraction of our muscles. So it's really, really important. So we're constantly using this as storage and then withdrawing from that storage, essentially. And so you could look at it kind of like a bank, our body's mineral bank. We can deposit minerals here, we can withdraw them. And if the bone is a bank, vitamin D is kind of like the banker. So your vitamin D levels in the creation of mature vitamin D is what is going to activate or deactivate those things. Okay. So this might be a good place to stop. I guess we have 20 minutes. I want to make the use of all of our class time, but also want to have stuff to talk about on Thursday and don't want to overload you guys. I think I'm going to continue. And we'll have some activity/review of this stuff on Thursday, since it looks like we're going to be able to get through all of it today, which is good. So we went over our most diverse and most complicated tissue types from this chapter. We did epithelium and connective tissue. And so the rest of this is just going to be looking at the different types of membranes along our tissue. So we have 4 different types of tissue membranes, and then a brief look at muscular tissue, muscle tissue, and nervous tissue. So essentially, we have 4 different membrane types lining our tissues. We have mucous, serous, cutaneous, and synovial. And some of these are really specific, or I guess all of these are generally quite specific here. So mucous membranes secrete mucus. Serous membranes secrete serous fluid, and I'll talk more about like what that is and what they do. The cutaneous membrane is what makes up the external portion of our skin. So it's just a whole bunch of those stratified layers of squamous epithelium. So there's lots of layers to be rubbed off and resist that friction without allowing damage to the connective tissue below it, which is where your blood vessels and nerves are. And then we have synovial membranes, which are only present within synovial cavities, within synovial joints. And so these are going to be all the joints where the most amount of movement is possible. And so we'll get back to that when we are looking at that specific slide. So the first example here are mucous membranes. So mucus is essentially just protection from whatever is inside that environment that it is being secreted into. Right? So we have mucus that goes into that gets secreted throughout our throat and respiratory system and nose to protect against stuff that we are breathing in. We have mucus that is secreted into our stomach lining to protect against the acid that we secrete into that stomach and resist damage to the cells. And so, yeah, these are there to protect in those areas. So found in the digestive and respiratory tracts, as well as the urinary and reproductive tracts. We then have serous membranes, which is what lines all of the ventral cavities. So I guess to try to simplify this even further, mucous membranes will typically be found in like cavities within organs. That's too small. I'm gonna add that in. So mucus is typically secreted directly into cavities within the organs, whereas serous fluid is secreted into cavities between organs, like cavities outside of that organ, essentially. Why? Take a long time to find. Right. And those are our ventral cavities. If you use that term ventral body cavities, that's referring to the cavities where we have organs interacting with other organs. And so that serous fluid is kind of what's lining that area, compared to mucus being within that organ itself, again, like the stomach and respiratory system. This cutaneous membrane, again, is just referring to the outer surface of skin, right? These highly compacted, stratified squamous epithelium. So lots and lots of layers of those biologically inexpensive, really thin cells. And then lastly, our synovial membranes. So we have a whole chapter on joints where we'll be looking at the different types of joints, but synovial joints are the joints where there is the most amount of movement available in that area. So shoulders, knees, etc., elbows, and that is because of this synovial fluid that gets secreted by the synovial membrane. So we have this fluid-filled sac between the cartilage that is lining these bones where they would potentially touch. And that fluid allows for some more mobility, a bit more shock absorption, etc. And so that is what synovial membranes secrete synovial fluid within that space, essentially. Okay. So our last two major tissue types. Again, we'll go into much more depth about both of these throughout this course, but we'll briefly address them here as far as the types or some of the characteristics. So we have three different types of muscle tissue. Skeletal muscle, which is typically connecting to the skeleton. Cardiac muscle tissue, which is only found in the heart, only found in the heart, and then smooth muscle tissue, which is lining some of our other organs, essentially. So skeletal muscle tissue is voluntary muscle tissue, meaning you have control over it. It is the only voluntary muscle tissue here. Right? So these are going to be involved with movement. It's also involved with things like breathing and blinking, where you don't need to think about those things in order to control it. Your body will naturally, those are automatic muscle tissue. Your body will automatically contract and expand those muscles to allow you to breathe and to blink without needing to think. But you can control them if you want to, if you think about it. You can control the muscles of your diaphragm, control the muscles that control your eyelids to hold them shut or hold them open, or to take a big breath in and big breath out, right? But you don't need to. So those are automatic, but they are technically voluntary, meaning we can control them with neural signals. So some characteristics to look for that identify skeletal muscle tissue besides the function. They are really, really long. Each muscle fiber, each muscle tissue extends the entire length of that fiber. So very, very long, some of the largest cells in the body besides the ovum and our neurons. But very long cells. They have multiple nuclei So if you look at a stained muscle cell under a microscope, you'll see a few of those dark splotches, those nuclei. So these are multiple nuclei within one cell, just because it is so long, right? We have multiple of those control centers throughout. And they have these striations, which you'll get to understand better when we look at how a muscle cell actually functions and contracts. But those are just alternating muscle fibers, essentially, that make our contractile units that allow them to contract. So if you see long parallel cells like this with striations and multiple nuclei, you are looking at skeletal muscle cells, skeletal muscle tissue. Because these are doing such large contractions and utilize a whole lot of energy, they also typically have a high concentration of mitochondria. But yeah, so they extend the entire length. They do not branch. They are parallel, and they have striations with multiple nuclei. They are the only example here with multiple nuclei. Our cardiac muscle and our smooth muscle are both uninucleate, meaning they only have one nuclei per cell. Cardiac muscle tissue is only found in the heart. It is subconscious. It is automatic. It is you cannot control how your heart is beating through your neural signals. And it is unique with a few things. We have those striations again, like we saw with skeletal muscle tissue. But these cells do not run the entire length of whatever fiber or wherever they're connecting. We have separate cells, and they connect with each other at these intercalated discs. So they have very distinct connections. You can see the dark stain here, creating these intercalated fibrous discs holding those muscle cells together. And they have one nucleus, and they branch. So they're not exactly parallel or anti-parallel because they branch, they take different angles. But there's not a whole lot of overlapping of this muscle. And again, this is only found in the heart. So they join end to end at those intercalating discs, they have gaps and tight junctions, and they branch. Whereas the skeletal muscle did not branch. They're completely parallel. The one cell runs the entire length of the fiber. And then our last example is smooth muscle tissue. This is also involuntary. We cannot control it through thinking. And so this is what is lining all of our blood vessels to help allow some stretch and some contraction. This is what is surrounding some of your organs, like your stomach, that allow for the stomach to stretch and contract and move your food to digest within all of the hydrochloric acid and some of the gastric enzymes that are in there. Right? And so the structure of this tissue is unique in that they are overlapping. So not necessarily branching, but they do overlap. There are no striations. This is the only example here where we don't see those distinct lines throughout the tissue. And they are also uninucleate, so only one nucleus, and loosely packed, but not as structural as our other two were. And that's basically all we need for now on muscle tissue before we get into that actual chapter. And so the last thing here is our few slides on nervous tissue, which we will have multiple chapters looking into. I guess one chapter on the actual tissue and then multiple chapters on the nervous system. But essentially, nervous tissue is composed of neural cells and neurons and glial cells, ganglia. So the neurons are the cells that are actually conducting the signals. And then our ganglia or glial cells are what are providing all the support regulation as well as insulation. We do insulate the axons of our neurons to help conduct those electrical signals a bit faster. And so we're going to look at general structure and some of the names of the different types. So we do have a separation of our nervous systems. We have our central nervous system, which is our brain and spinal cord. And then the peripheral nervous system is all the nerves that extend throughout the body. Okay? And they communicate together and work together, obviously, but because we do separate them by system and have slightly different types of cells that exist within those systems. So the glial cells of the central nervous system, the CNS, are astrocytes, oligodendrocytes, microglia, and ependymal cells. Not going into depth about the structure and function of these just yet, but just introducing you to them. And then in the peripheral nervous system, it's just our Schwann cells. Our glial cells of the peripheral nervous system are these Schwann cells. And so basically what you will need from this portion of the chapter for this unit is to be able to recognize the general structure of a neuron, which is what we have shown here. So besides the ovum in females, or the egg cell, which is the largest cell in the human body, neurons are the second largest, essentially, or the largest in males. They are super, super long, or can be super, super long. And they all have the same general structure. We have the body of the cell, which if we cut, I'm just about to do this on my screen, like you guys can see what I'm doing with my hand. If we cut this portion off, right, it would look like a typical cell. We have kind of our globular structure with all of our organelles within it. And so this is the body or the soma of the cell, where you would find the nucleus, find all the major organelles. And the soma has these hair-like extensions called dendrites. And so this is essentially the receiving end of our neuron. So if the nucleus is the brain, right, we are receiving information right next to the brain. And then any information that's going to be sent out of this neuron needs to travel down the axon, which is this long tail-looking thing here, until we reach what will be the synapse. So the synapse is the space between neighboring neurons. So we have our cell body with all the major organelles. We have dendrites, which are named after tree branches, essentially, and they look like tree branches. And then we have our long axon, which is where the signal is going to be conducted. So it receives information from the dendrites, from the previous neuron, and then we'll send that information down its axon to the next one, sending signals throughout the entire body and receiving signals, sending them back to the brain from the entire body. And that is generally all you need to know here as far as the structure of an axon. And those are the only specifics about nervous tissue for this chapter. So yeah, that was a lot. It's the whole chapter. If there are any questions or concerns or topics you would like me to touch on again in these last few minutes here? Okay. I do want to point out really quickly some things on Canvas. Let me get these out of here. Are you guys seeing Canvas right now instead of my PowerPoint? Okay, cool. So first off, like I said I would do last week, I did record myself going over the chem and cell bio activity. I uploaded this in the cell bio video lectures. So if you're looking for that review, essentially I made a copy of the slide deck that you guys had worked on, made some edits and additions in red, so you could see what I added or changed, and then talked about kind of what a full answer to those questions as a written response question would look like. All of the pieces that you could have possibly included. It was generally what I was trying to provide. And so I discussed it that way. So the original slide deck that you guys can access still through that same link does not have my additions. So you are welcome to go in there and add on to it. That link is here. I think I also put it in the assignment that I posted so I could put in a grade. You're welcome to go add on to that. But at the very least, even if you're going to copy exactly what I wrote down, I at the very least wanted a student or multiple students to be doing the actual typing, thus generating some neural connection and hopefully increasing the actual learning of that moment rather than just giving you the answers that I would have been looking for. So that's available. I also wanted to point out this crash course series. So Crash Course Biology has a subsection with Crash Course Anatomy and Physiology. Some of you may already watch some of these videos. But their series on tissues in particular, I think is really, really helpful since there are so many specifics in here. The greens do a great job with everything they do. They're really awesome. I've been watching Crash Course for a long time, but I highly recommend you check those out. I just added that this morning, having thought about it. So there's 4 videos in that series. They're all like 10 minutes each, but check that out. Very, very helpful. And I said in lab not to expect that I'm able to find the time to make these PowerPoints every week, but I was able to put together a full appendicular skeleton PowerPoint. So we do have that for this week and next week too, to look forward to. Other than that, you have your little snapshot of the week and your assignments on the homepage. So please make sure you're taking care of those assignments if you want the points for them. Hopefully at this point, you're starting to get an idea of which assignment types are most helpful for you and which ones you want to dedicate your time to. But yeah, all the ones that are coming up with the those due dates are shown there. So any last minute questions, concerns, or comments before I release you back into the wild to endure the cold weather of Seattle, Washington? Okay. Well, thank you guys for coming today. I hope you have a great afternoon, and I will see you tomorrow to look at the pectoral girdle and bones of the upper limbs.
Thank you. Sorry. I have a quick question. Like, what are we expecting for the exams? Do you have like give us a general ideas what
Sure.
we can expect for the exams, please?
Okay, like, are you asking what content is going to be on the exam, or are you asking for like practice questions?
The contents.
Okay. So yeah, this is described in the syllabus as far as the separation here. Are you guys seeing Canvas again? So I just need to double check. Yeah, okay. But essentially the course is separated into units. We have our 3 units. Unit 1 is modules 1 through 4. So I guess the rest of this stuff isn't published for you to see it, but you do have the exam 1 module here. So basically everything before the exam 1 module is content for exam 1. So the lecture content is going to be chemistry, cell biology, and tissues. Right, there was another chapter in here. No. Cool. So yeah, chapters 2, 3, and 4 from the textbook for lecture content. And then there is a separate exam for lab content, more of a practical, which is going to be on the bones of the skeleton. And so every week there's that lab info for the module. Those are the bones, markings, and any additional information. So I guess this one's just directions and planes. It's not a great example. Additional information, meaning like which other bones it articulates with, that will all be identified as far as what you're responsible for in these lists, or if it's a left or a right bone. Okay. So for the vertebrae, being able to name a bone or be able to find the bone based on the name. Same thing is going to go for our phalanges and metacarpals and metatarsals, right? Those have names, all the specific bone markings. So we're not covering every single bone marking, or I'm not asking you to memorize every single bone marking, but the ones that you are responsible for are listed here. And then let me pull up the one from module 4, because that has some bones that you are being asked to recognize left versus right for. So like a femur, if you were given a femur to analyze, it's very likely I'm going to ask if is this a left or a right femur, or an os coxa, or any of these large bones, a scapula. So those that have relatively clear distinguishing marks that would determine whether it's a left or a right bone, you could be asked to determine that. Give me that. So yeah, does that address your question?
Yes, thank you.
Okay, yeah. And so the lecture exam is going to be a mix of question types, some multiple choice matching type stuff like you see in the quizzes, and then a few written response questions like what we did in that activity the other day. So what that slide deck shows. And then the lab exam is much more of a practical. You're given a sample, a model, and asked to identify it or identify the pieces of it based on what's in front of you.
Thank you.
No problem. And I guess, I don't know if you intentionally waited until everyone left, but that is a good question for everyone to have heard. So yeah, please ask questions like that while everyone's together too.